I will start with a short introduction to lecture 13. Biomedical engineers who work in the field of implantable materials and devices must always keep in mind a couple of important facts. Specifically, that whenever that implantable material and device is introduced into the body, it requires a surgical procedure which inevitably brings about injury to cells, tissues, and organs. Furthermore, the presence of the implant material and device forms an interface at the tissue injury side. On one side is the physiological environment containing cells, extracellular matrix, biological fluids, blood vessels, and nerves, but on the other side is the synthetic material and the synthetic implantable device. Taking a closer look at what surrounds that particular interface is shown on this particular cartoon. On one side, we have the biological physiological environment, and on the other is now the biological physiological environment, which includes the synthetic biomaterial out of which an implantable device is composed. We see on both sides of this particular interface certain similarities, specifically the cells, the biochemical, biophysical stimuli, blood vessels, nerves. What is the big difference is this middle part. In the biological physiological environment, we have the extracellular matrix of the tissues. On the other side, in the environment around the implants, we have the synthetic biomaterial, which may be a ceramic, a metal, a plastic, or a composite of any one of two or three of these aforementioned types of materials. For this reason, a critical requirement for implant materials and devices is that the materials which are used for that particular purpose must imitate, duplicate, simulate the extracellular matrix of the physiological tissues. This is the only way that the synthetic material and device will provide an environment to the cells, the living component of the tissues, and in order for them to survive, to function, and to form new tissue. This requirement has led biomedical biomaterials engineers into the following conclusion. In order to have biocompatible materials and therefore devices composed of those materials, we have to learn from physiology and biology the best that we can regarding the extracellular matrix, which is now replaced by our favorite biomaterials. Learning what is the composition of the extracellular matrix, how it functions, and translating that information into the synthetic biomaterials which most biomedical engineers are working with is a great requirement, and it is an approach which has been followed by biomedical engineers presently, and it is an area in which I expect that we will see continued work and many more contributions in the future. The sources of information and inspiration which biomedical engineers have used in order to address the requirements I just summarized have been now cellular molecular biology, specifically protein and cell interactions with substrates, and the substrates could be either natural or synthetic. This brings us to lecture 15A. The title of this lecture is A Short Introduction to Proteins. Please note that all the figures and tables of this lecture are from Chapter 2 in the course textbook. Let us start by reviewing some important aspects, I will call them facts, which are pertinent to the topic of this chapter. First and foremost, the fluids of the human body, for example, blood, lymphatic fluids, urine, etc., all of them contain proteins. When we reviewed the blood plasma, the liquid part of our blood, we mentioned that it contains dissolved coagulation proteins, complement proteins, and immunoglobulins. In addition, the human tissue, specifically their extracellular matrices, contain proteins and proteoglycans, which are some form of proteins. Examples of such proteins, of course, first and foremost, collagen, ubiquitous, different types of collagen, but found in many tissues in the body. Other protein examples are fibronectin and laminin. Some examples of proteoglycans are aggregan, decorin, syndecan, etc. Why should biomedical engineers be interested in proteins? This is the very important fact. Within seconds following the introduction to the human body of implantable materials and devices, they are covered by adsorbed proteins. Adsorption means that the proteins are adhering on the surface of the material. We need to make a distinction in the terminology. Adhesion is used for cells. Adsorption is used to indicate the interaction of proteins on the surfaces of synthetic materials. Another important reason for which biomedical engineers must know what is happening with proteins is the fact that proteins mediate the interactions of cells with substrates and also mediate cell functions pertinent to new tissue formation. And this is the part of the wound healing process which was covered in the second and part of the process, actually the third and the fourth stage, proliferation and repair, and remodeling and maturation. We'll start with a review of protein composition and structure. Since some of these aspects have been covered in other courses which you, the students in the biocompatibility of materials course, have already taken, specifically the cellular engineering, we are not going to spend too much time on these particular topics. Suffice to say, and let it be a good reminder, that amino acids, a total of 20, are the building blocks of proteins. It is a fascinating aspect that the smallest components of the structure of proteins, which are so important in biology and physiology, are 20 different amino acids. Table 2.1 from the chapter number 2 from the textbook gives us a list of all known amino acids. You have in the column on the left the full name of each one of these amino acids. For example, alanine, glycine, arginine, etc. In the middle column, you have an abbreviation which is used in the field, and it involves the first 3 letters of the name of each amino acid. For example, in the case of alanine, the first 3 letters are ALA. The third column, the one on the right, has a 1-letter abbreviation for the names of each amino acid. In some cases, you'll see a correspondence between the first letter in the name of the amino acid and the one letter abbreviated notation. Specifically, alanine starts with an A. The one letter notation for this particular amino acid is A. This is not the case for all amino acids. For example, aspartic acid starts with A, but the one letter denomination is the letter D. In the case of arginine, another amino acid which starts with the letter A, the one letter notation is the letter R, and so on and so forth. Therefore, if you are going to use the one letter notation for an amino acid, you have to learn the correct one letter notation for each of the amino acids. Otherwise, I will suggest that you use the full name of the amino acid. Amino acids are organic compounds. Therefore, they have a carbon or more than one carbon atoms in their core. In figure 2.1, you see schematically in the chemical scheme the general structure of an amino acid. You see the carbon atom in the center, and then because of the 4 valences, you see the capability of the carbon atom to interact with 4 more atoms. In the case of amino acids, the carbon atom interacts and makes a bond with hydrogen, the amine group, and the carboxyl group, COOH, the amine NH2. The 4th one is always indicated by R, and it can be different for the different types of amino acids. In figure 2.2 on this particular slide, you see an important aspect, namely that depending on what kind of an environment, specifically the pH of the environment, an amino acid finds itself, it can have different charge from positive to negative. And this is a very important aspect to keep in mind when you are dealing with amino acids. The simplest structure is one carbon atom in the center attached to 4 other atoms. Actually, most of the amino acids are what we call polypeptides. That means that we have many more units, therefore many more carbon atoms in the core. Figure 2.5 from the textbook illustrates the 2 processes which give us this polypeptide outcome. In part A of this figure, you see a schematic of the so-called condensation reaction. And what is shown over here is that we have 2 of these units of a particular structure with 2 carbon atoms joining and forming a longer unit, and during that particular reaction we have the release of one water molecule. In the case B, you see an example of what is called sequential addition of amino acids. So one amino acid is attached to another, and we have, as you can see, a very long chain, which is of course referred to as a polypeptide. This is definitely a busy slide, figure 2.3 from the textbook, which now not only show us the chemical structure of all 20 common amino acids, but groups them along the following categories: non-polar amino acids, polar amino acids, negatively charged and positively charged amino acids. Please refer to this table whenever you need to elucidate the specific structure of a particular amino acid and or confirm the polarity and the negative or positive charge of a respective amino acid. We mentioned that The amino acids are the building blocks of proteins, and figure 2.6 from the textbook gives a schematic illustration of how we get that particular formation of a protein, starting with the building blocks, the amino acids. The first part of the figure, entitled primary structure, shows now a string of amino acids connected with each other. This particular structure now can loop around in itself, and then we get the secondary structure with these interesting loops, still relatively simple. In the case of tertiary structure and the quaternary, you can see that these particular loops can get more involved and more convoluted. And these are the proteins. Definitely proteins are big, molecular weight-wise chemical compounds, and they have a very involved, convoluted structure, which is schematically shown in this figure. Figure 215 from the textbook illustrates the structure of a protein which is ubiquitous. It's found different types, but it's found in many tissues and organs in the body. Collagen. Starting with the first illustration, a reminder that each protein, and definitely the amino acids, they have an N-terminal which is associated with NH2 ending, and a C-terminal which is associated with the carboxyl COOH ending. Collagen has 3 strands which are interconnected, and depending on what type of microscopy is used to examine the structure, you have the illustrations in frame C and in frame D. I will make some comments regarding frame D, because when histological examination of various tissues was studied, they noticed in the extracellular matrix this particular, which sometimes colloquially they call it railroad arrangement. You can see this particular parallel black interchange with white bars, and this is an indication of collagen fibrils, which are a result of the way collagen structure is staggered. And this is evidence of the presence of collagen in the extracellular matrix of many tissues. Another example, which is illustrated in figure 217 from the textbook, is elastin. Elastin is a protein which of course has mechanical properties, and one of the properties is that it can be stretched. And here is a schematic illustration of what happens to the structure of these molecules when the tissue is relaxed. You see all these kinds of googling structures, but when the tissue is stretched, you can see this alignment of the elastin units and some kind of linear kind of arrangement. A third example from figure 219 in the textbook is fibronectin. Fibronectin is very important because it was one of the proteins which was studied extensively, and as a result of that, the mechanism of how cells interact with proteins in the extracellular matrix was discovered. In this schematic illustration, you see the N-terminal NH2 on the left-hand side of the figure, and the carboxyl, the COOH ending of the protein, on the right-hand side. The circles in between represent each one of them either an amino acid or a group of amino acids. For example, in this particular figure, what is indicated as RGD stands for arginine glycine aspartic acid, is one of the circles, but it involves 3 amino acids. In addition, when this protein was studied, people identified certain domains, which of course they labeled appropriately fibrin heparin or collagen gelatin or cell binding domain, the domain that binds heparin, or fibrin. These are very important aspects which were discovered as a result of very beautiful research conducted by colleagues in cell biology and biochemistry. The way these tests were conducted was to use somehow chemical scissors. They're not real scissors, but they're chemical compounds, enzymes specifically, which can start from one end of the protein and start cutting or cleaving certain domains, connect all those domains and test them for bioactivity. And of course, the way those collected segments were tested was to check their bioactivity in terms of promoting certain cell functions. And of course, the cells were chosen very judiciously among the cells of the tissue in which this particular protein, fibronectin, is found in the human body. Elucidation of the composition, the amino acid composition of various proteins, and identification of the various sections of the protein which have biological activity has led to the following list of functionality of regions and then peptides. Peptide is a name which is given to a group of amino acids. Celebrated among them is, of course, the tripeptide arginine glycine aspartic acid, with the abbreviation of one letter per amino acid, RGD. This particular peptide was first identified as a result of the research with fibronectin, and it turned out to be the region in the protein which the cells identify and they adhere. Keep in mind that proteins are big, bulky molecules. This research provided evidence that the cells do not interact with the entire bulky, big protein molecule. They recognize only specific domains, also known as epitopes. For example, the RGD, 3 amino acid unit, with which they interact using other proteins which are called receptors on their cell membrane. You can see that different cell adhesion domains have been identified for various cells. For example, the cell-cell adhesion domain that is GRALARGEANF, a mouthful, yes, is a neural cell adhesion molecule. We have other details which might be of interest to you if you are doing appropriate research. The important thing, and that's why RGD has been celebrated, is the following. RGD was discovered when researchers worked with fibronectin, but then it turned out that it is a 3 amino acid sequence which is found in many proteins, for example, collagen and other ones of the extracellular matrix, and it has become a celebrated domain or epitope of proteins which the cells recognize with their membrane receptors and interact with them adhesively. The domains or epitopes of the proteins, in addition, are referred to as ligands. I will close this particular lecture 15A by reminding you, as a result of this research pertinent to amino acids, it was discovered that proteins mediate the interactions of cells with substrates, and they promote cell functions which are pertinent to new tissue formation. Those examples of cell functions include cell adhesion, migration, cell proliferation, etc. As a result of pertinent and related research, the present and future requirements for biomaterials, implantable biomaterials and devices, are that they must be not only biocompatible, but they must replicate the biological, physiological environment of the target cells, tissues, and organs. And this is the environment in which we, as biomedical engineers, specializing in implantable materials and devices, work, and our products are reviewed, evaluated, and are considered successful if they meet these two criteria. This is the end of lecture 15A.